one is the most virulent? Pardon?
Second.
The what?
Cutaneous.
Yes, the cutaneous. The fewer it takes to infect half of the sample population, the more virulent that organism is. So that's what you're looking for. And there's going to be a test question on that, so make sure you know that. The LD50 or lethal dose is how many bacteria or virions are needed to kill 50% of the sample population. They also use this LD50 to talk about the potency of a toxin. So for botulism toxin in mice, the LD50, oops, I made the 50 go on the top, down, is 0.03 nanograms per kilogram. For the Shiga toxin, the LD50 is 250 nanograms per kilogram. So which one of those is more potent? The botulism or the Shiga toxin?
Botulism.
The botulism. So we've got the organisms inside of our body or on our body. We've already talked about portals of entrance. Now the pathogen has to attach or adhere to the host. And they attach to the host cells by adhesions. And these are projections on the surface of a bacteria that adhere to complementary receptor sites on the host cells. And adhesions bacteria has fimbriae on it to help with adhering to tissues. And on the ends, they have what's called an adhesion. And these adhesions on a host cell, they'll be a receptor site that will line, that is exactly a perfect match for that adhesion of the pathogen. So adhesions are associated with fimbriae, with the glycocalyx, which is the could be the capsule or the slime layer, the flagella. For the helmet, they use hooks, barbs, and suckers to attach to their host. We talked about the tapeworm. It has the scolex with the suckers and the hooks on it that attach to your intestines. After the bacteria adhere to you or stick to you, then that's followed by colonization of the tissues and may involve invasion of the cells. An example of this colonization is the formation of biofilms. The microbes secrete a glycocalyx, and then that helps those bacteria to stick to the host cell, and it helps the bacteria stick to each other, so it forms this film. This allows the bacteria to share nutrients, and it also makes that biofilm makes it so they are less likely to be able to be affected by your immune cells or by any type of antimicrobials that you would be taking. And a bacteria that forms a biofilm is called Corynebacterium diphtheriae, and that's the one that causes diphtheria. And diphtheria, those bacteria grow on the back of the throat and form a biofilm there, which can actually get quite thick. So that's just showing the adhesions on the bacteria, and you can see the little receptor sites on the host cell membrane where it lines up perfectly. Penetrating, now step 3 is penetrating and surviving the host defenses. One way they can do that, and this would make a bacteria virulent, is if they have a capsule. So if they have a capsule, it makes them resistant to phagocytosis or by your immune cells. Because your immune cells usually go out and take this bacteria inside of them and eventually break it down into pieces. But with the capsule, they avoid that. Streptococcus pneumoniae causes pneumococcal pneumonia, and to cause pneumococcal pneumonia, those bacteria have to have a capsule. That makes them virulent. Those that don't have a capsule don't cause the pneumonia. So here's a picture of bacteria with a capsule. And you can see it would be harder with that around them for antibiotics and immune cells to penetrate them. So there are some other cell wall components that help bacteria avoid phagocytosis, and it aids in adherence to the host cells. One of them is, remember when you did the acid fasting? Why were we doing that? Who was the bacteria?
Because they have a liquid around, they have liquid around, they into the gram snake. They contribute up with gram snake because
Yeah, they clump together because they have this sticky substance in their cell walls called mycolic acid. And it makes it hard for things to penetrate that. So it would make it hard for antibiotics to penetrate it and your immune cells to get in there. And we know that it's hard to penetrate because we had to heat it up for 5 minutes to drive the dye through those cell walls. We couldn't do a regular stain like a gram stain on the acid-fast organisms. Another cell wall component is M protein. It's heat and acid resistant, and it's produced by Streptococcus pyogenes. And it aids in attachment to epithelial cells and resistance to phagocytosis from the white blood cells. Enzymes, extracellular digestive enzymes are able to lyse cells and to dissolve the material between the cells and to form blood clots. So this is a virulence factor for the bacteria. One type of enzyme are called coagulases. They're produced by some of the streptococci, and they convert soluble fibrinogen, that's circulating in your blood, to an insoluble fibrin. These are kind of threads, and they form a blood clot. And then the clots, because the bacteria now are inside that clot, and that clot protects the bacteria against your immune cells, so your immune cells can't go out and eat them. Phagocytosis means cell eating, and other parts of the immune system. They're protected inside that clot. Kinases are produced by Staphylococcus and Streptococcus. That enzyme digests the fibrin and dissolves the blood clots. So sometimes when you have an infection, your own body starts to form a kind of a clot around your infection, and the the bacteria have that enzyme that can dissolve that clot, and then it frees the bacteria to spread to other parts of your body. We use that enzyme, streptokinase, that is produced by these two bacteria. We use it if somebody's had a heart attack, then they get an injection of streptokinase if it's right away, if they catch it quick enough. Another enzyme is hyaluronidase, called the spreading factor. And it is produced by some streptococcus and by Clostridium. So if that enzyme digests somebody pronounce that for me. That acid, anyway, that acid holds cells together and in connected tissues, and the enzyme then can break those cells apart. Collagenase is produced by Clostridium, different species of Clostridium. It dissolves the collagen framework of muscles, and then that helps aid in the spread of gas gangrene. That is something that is caused by Clostridium. Then some bacteria produce IgA proteases, and maybe next week we'll get into the antibodies, but one of them is IgA. And these proteases then destroy those antibodies. And some viruses and bacteria have what's called antigenic variation. And that's where microbes are able to change their surface antigens. And when you have an organism, the antigen can be on the surface, on the fimbriae or whatever, and your immune system comes out, recognizes that antigen, and starts making antibodies specifically against that antigen. So your body starts fighting this disease. And with influenza virus, after a while, that influenza virus changes those antigens. So now your antibodies aren't able to work against that influenza body. So then it has to make new antibodies. And this is why when you need to get a new influenza or flu shot every year, is because the virus changes its surface antigen. Siderophores. The pathogens need to use some of the host nutrients. They need iron to grow, and inside the host cell, the iron is usually tightly bound to iron transport proteins such as hemoglobin. So some microbes, since they need that iron, they secrete proteins called siderophores, and these can take the iron away from the iron transport protein and form an iron siderophore complex. So it can take the iron from the host cell, make a complex that this pathogen is going to be able to use. So then these siderophore, these complexes bind with the siderophore receptors on the bacterial surface, and then the iron can be brought into the bacterium either as that whole complex, or it'll just take the free iron off of it. So when a cell lyses, that also can release pathogens. So pathogens, part of the, especially a virus, it